Thank you to the organizers for the kind invitation. During my presentation, I would like to show to the audience my most recent data on tissue transglutaminase and its interaction with integrin-linked kinase, which have been recently published, and how this interaction is strictly correlated with the progression. Ovarian cancer is relatively uncommon, yet is one of the deadliest cancers. Over 250,000 women are diagnosed with epithelial ovarian cancer each year in the world, and with almost 22 cases just in the United States, and unfortunately more than 14,000 women die from this disease in the United States. Ovarian cancer has been called a silent killer because no distinctive symptoms have been associated with the early stages of this disease, and most women with ovarian cancer report symptoms or non-specific gastrointestinal complaints, and many women recall having had the symptoms for an extended period of time before diagnosis, until the symptoms unfortunately worsen, and at this point the cancer is often widespread and difficult to cure. Current standard treatment for ovarian cancer in both early and advanced stages consists of complete cytoreductive surgery followed by chemotherapy, usually based on a platinum and taxane combination. If we see this graphical representation, we put the level of the ovarian cancer-related tumor marker, CA 125, across the disease treatment course of the patient diagnosed with ovarian cancer. The graphical representation shows that the initial response rate is high. Almost 70 to 80% of patients respond to the first treatment, but unfortunately, the majority of patients with advanced disease relapse within the first 2 years after diagnosis, which results in a cycle of repeated surgeries and additional rounds of chemotherapy. And as we can see, the relapse rate is correlated with the stage of the disease. Advanced stage, like 3 and 4, correlate with, unfortunately, very bad overall survival and relapse rate. So the identification of a possible target and successful therapies is needed. This reason, a few years ago, we studied a large cohort of ovarian tumors, 93 to be specific, and we analyzed this cohort for the expression of a tissue transglutaminase type 2 or TG2. We saw that 73% of the tumor scored medium to very high TG2 staining. And the staining was strictly correlated with the histological grade. Stage 3 and stage 4 showed very strong staining. On top of that, we analyzed the TCGA ovarian cancer database, and we saw that high expression of transglutaminase was significantly correlated with a worse overall survival in a patient compared to patients that have less tissue transglutaminase expression. But what is transglutaminase? Transglutaminase is a multifunctional protein which can be secreted in the extracellular compartment and also works in the intracellular compartment. TG2 has 4 distinct domains: an N-terminal beta sandwich domain, which shows, has been shown that promotes TG2 interaction with fibronectin and integrin, promoting integrin-mediated binding of cells to the extracellular matrix. Tissue transglutaminase also works as an enzyme, as a catalytic core, and promotes post-translational modification of proteins in presence of calcium. Transglutaminase is also associated with the G proteins, where in presence of GTP, works as a GTPase and is mostly involved in signal transduction. These multiple functions are regulated by its cellular concentration, intracellular or extracellular distribution, as well as the presence of the cofactors calcium or GTP. In the presence of calcium, TG2 has an open conformation and catalyzes the calcium-dependent cross-links of a protein with a glutamine residue called a glutamine donor to another protein with a lysine residue, which is called the glutamine acceptor, resulting in the formation of an epsilon gamma glutamine lysine isopeptidyl bond. TG2 also catalyzes the dependent incorporation of primary amines, such as polyamines and histamines, into the protein, which is called, this process is called transamidation. TG2, in the presence of GTP, acquires a closed conformation where the catalytic core is blocked and hydrolyzes GTP to GDP, and in association with PLC gamma promotes intracellular signaling. Growing evidence indicate the key role of tissue transglutaminase in the extracellular matrix reorganization and as a mediator of outside-in signaling. The extracellular transglutaminase and this interaction with fibronectin and integrins promotes cell adhesion and cell matrix interaction. It has been also shown that transglutaminase, by binding fibronectin, can also interact with other tyrosine receptor kinase, Wnt receptors, and other ligands to promote an outside-in signaling. And the aberrant activation of this outside-in signaling has been correlated in several cancers, and also we studied in ovarian cancer with epithelial to mesenchymal transition, invasion, metastasis, and stem cell-like phenotype. As alteration in the level of transglutaminase expression modulates epithelial ovarian cancer cell adhesion in vitro and in vivo, we examined whether TG2 interacts with integrins and fibronectin in ovarian cancer cells. Immunoprecipitation with fibronectin and beta-1 integrin beta-1 antibodies followed by immunoblotting for TG2 shows endogenous interaction between TG2 and fibronectin and TG2 and integrin beta-1. Most importantly, TG2 and beta-integrin and TG2 and fibronectin complexes are enriched in ovarian cancer cells, as shown here by immunofluorescence and confocal analysis, giving a functional significance in ovarian cancer. So our next step was to understand how the interaction, the extracellular that occur in the extracellular matrix between fibronectin, TG2, and integrins translate into the activation of outside-in signaling. And we set out to study the first kinase, which is linked with the integrin beta-1 cytoplasmic domain, which is the integrin-linked kinase. Integrin-linked kinase is a scaffold protein which has both scaffold properties and also works as an enzyme, as a kinase. ALK, as you see, localizes to focal adhesion, where function as a kinase, adapter proteins, and couples integrins and growth factor receptors to the actin cytoskeleton and the cellular signaling events. So by interacting with these proteins, integrin-linked kinase has been shown that regulates several cellular processes, so increases the formation of focal adhesions, stress fibers, and activation of several growth factor receptors and tyrosine receptor kinases, which translate in cell migration, cell invasion, and cell survival. So, having seen that transglutaminase interacts with fibronectin and integrins, and knowing that integrin-linked kinase directly interacts with the beta-1 integrins and activates intracellular signaling, both survival through AKT signaling and others through proliferation by phosphorylating and inactivating GSK, we set out to understand whether transglutaminase may promote the outside-in signaling by regulating integrin-linked kinase and activating some the ovarian cancer progression. To do so, we plated the ovarian cancer cells on fibronectin matrix and show that there is a sustained activation of integrin-linked kinase through its phosphorylation as residue, which is serine 246. When tissue transglutaminase was specifically knocked down, we saw that there is a significant decrease in the phosphoactive ILK in two different cell lines, SQV3 and OVCAR5 cells. So next step was to understand if these two proteins, they may come in cluster. So we saw that in the presence of transglutaminase, there is an enrichment in the active phospho-LK, and when tissue transglutaminase was knocked down, we saw a significant decrease in this signal. But most importantly, through confocal analysis, we found that there is a strong interaction between these two proteins. The Pearson colocalization was 0.42, and 89% of the phospho-ILK volume colocalizes with transglutaminase. And of course, this possible interaction, this colocalization, was disrupted in the absence of tissue transglutaminase. So, to determine whether transglutaminase and active ILK complexes are detectable in human tumors, we used the proximity ligation assay, a technique capable of identifying proteins localized within 40 nanometers of distance in tissue by using pairs of affinity probes conjugated with secondary antibodies, fluorescent secondary antibodies. By PLA, we identified that TG2 and active ILK form complexes in human tumors identified by discrete dots, and this interaction was not occurring in the absence of fibronectin, identifying that the extracellular matrix rearrangement and was important in translating, in the presence of transglutaminase, the outside-in signaling through integrin-linked kinase. Most importantly, we found that tissue transglutaminase and ILK in the TCGA analysis show that patients that have increased tissue transglutaminase and ILK showed worse overall survival compared to patients that had the lower expression of these two proteins. So at this point, our interest was in identifying some molecular pathways correlated with the increase of tissue transglutaminase and the activation of integrin-linked kinase. In previous reports, we have demonstrated that in the context of cell matrix interaction, tissue transglutaminase, by interacting with fibronectin and integrins, specifically regulates beta-catenin signaling through cis-arc, modulates its phosphorylation, and disrupts the interaction with icadirin, disrupting cell-cell interaction. Beta-catenin at this point, freed through this ligand icadirin, goes into the nucleus and promotes cell proliferation and oncogenic progression. We saw that cells plated on fibronectin had an increase in beta-catenin, and when tissue transglutaminase and integrin beta-1 were decreased by using or inhibited by using inhibitory antibody, which is 4G3, which is an anti-inhibitory antibody for transglutaminase, and 2A1, which is an inhibitor antibody for integrin beta-1, we saw a decrease in beta-catenin. And the increase in beta-catenin was correlated, reversely correlated with icadirin expression. Increase in beta-catenin correlated with a decrease in icadirin. So we show that there is a potential mesenchymal epithelial to mesenchymal transition. So we better investigated this pathway, and we used the antibody, the clone 4G3, which is an inhibitor antibody that binds specifically and blocking the fibronectin binding domain in tissue transglutaminase, and we used the anti-beta-1 integrin inhibitor antibody, the clone P5D2. By treating cells with this antibody, we checked for self-survival, proliferation, and invasion. So to see if beta-catenin was indeed regulated by transglutaminase function, we plated the cells in the presence of beta-catenin Wnt signaling activation, which is Wnt3a, which activates the canonical Wnt signaling. And we saw that there is a sustained increase in the presence of Wnt3a of active focal adhesion kinase, phospho-Fc, and a significant increase in ALK, active ALK. And in the presence of these two inhibitory antibodies, this activation was significantly disrupted. Most importantly, we saw that indeed, as expected, Wnt3a increased the active beta-catenin level, and at the same time, these two antibodies specifically blocked the beta-catenin activation. And we also demonstrated through immunofluorescence, we saw that in the presence of Wnt3a, active beta-catenin is mostly localized in the nuclei, and when we block specifically integrin beta-1 and Tg2, this nuclear translocation is decreased, as well as the transcriptional activation of beta-catenin and the expression of one of the genes regulated by beta-catenin, such as c-Myc. So next step was to investigate if indeed transglutaminase regulates beta-catenin through ALK. To do so, we used a specific inhibitor, a small molecule inhibitor for ALK, compound 22. And by treating cells with Wnt3a, we saw that indeed there is activation of FAC and ALK. And in the presence of ALK inhibitor, this activation is decreased. We also saw, confirming our theory, that there is an active beta-catenin, and this active beta-catenin in presence of Wnt3a, and the active beta-catenin was decreased in the presence of CPD22. We confirmed this data by immunofluorescence. We confirmed that in the presence of Wnt3a, we saw that there is a specific nuclear staining for active beta-catenin, but when we treat the cells in combination with ALK inhibitor, the Wnt signaling is disrupted. We didn't detect any active beta-catenin in the nuclei. We translated the study in the functional significance of a decrease of beta-catenin in the nuclei by using TCF-LEF luminescence activity, and we saw that in presence of Wnt3a, we have an increase in the transcriptional regulator, TCF4-LEF1 in the presence of Wnt3a, and this one was decreased in the presence of the ALK inhibitor. So, next step was to evaluate whether the Tg2 fibronectin-dependent activation of ALK has functional consequences. So we measured the effects of transglutaminase-mediated ALK engagement on activating the oncogenic Wnt pathway by assessing proliferation and invasiveness of ovarian cancer cells. We saw that Wnt3a treatment efficiently increased the proliferation and the migration in ovarian cancer cells compared to control. And 4G3 treatment drastically reduced the Wnt3a, induced both proliferation and migration. At the same time, to evaluate whether integrin-linked kinase mediated beta-catenin signal is implicated in the proliferation and migration of Tg2 expressing cells on fibronectin, We measured the same endpoints in cells in which ALK was inhibited pharmacologically by the inhibitor CPD22. As expected, Wnt3a increased both ovarian cancer cell proliferation, migration, and when the cells were treated with the ALK inhibitor CPD22, we saw a significant decrease in migration as well as in cell proliferation. We detected whether the ALK functional inhibition was implicated in tumor formation. So we divided the animals in 4 groups. Some were treated with carboplatin, others were treated with the ALK inhibitor in combination. We saw that ALK alone and most importantly in combination with carboplatin significantly decreased tumor volume and tumor weight, as well as decreased the number of metastases. So, concluding that most likely tissue transglutaminase, by interacting or reorganizing the extracellular matrix environment, translate the extracellular cues in the outside-in signaling through ALK. ALK, at this point, regulates the oncogenic Wnt signaling, promoting tumor progression. But when we block specifically integrin-linked kinase, we both block tumor progression, cell proliferation, and cell migration, possibly identifying a novel target that can be used for therapeutic. So in conclusion, we found that tissue transglutaminase directly interacts with and facilitates the phosphorylation and activation of ALK. This complex, TG2 and active ALK, are detectable in patient-derived primary cells. TG2 mediated ALK activation promotes beta-catenin nuclear translocation and transcriptional activity, and the inhibition of transglutaminase and ALK blocks cell addition to fibronectin matrix, Wnt response to the Wnt3a, and ultimately tumor formation and metastatic progression. I'd like to thank all my collaborators, of course my grant support, the Indiana University, of course the people that work in my lab, the present and the former, and you all for the kind attention, and I'm open for if you have questions. Thank you all.